Chalo, let's start our topic. So, today we are going to start our new chapter, Principle of Inheritance. As I told you in the last class also, this is very, very, very important as well as difficult chapter. Okay? So, this chapter requires your focus and full concentration. Okay, everyone? Now, look at here. As you can see, the image is related to the family, father, mother, and the son. Okay? As you can see in the image, father is having some specific characteristic like eye color, shape of ear, the height, right? Hair. Whereas you can see in the characteristic of mother, definite characteristic of hair, eye color, height, the shape or the size of the ear, right? So if you will see the characteristic or the traits, so it's better to use the word traits of the son. You will find that some of the characters, some of the traits are similar to the dad, some are similar to the mom. For example, eye color. Father and son, they both having same eye color, right? Hair. Mother and son, they both have a same hair type and color. Shape or the size of ear. The father and son, they both have the same type. Whereas if you will see the height of the son, you will find that the height of the son and mother is similar. So these traits or the similarity between the parents and the children shows what? Yes, Sarjana.
Ma'am, it's a show about how he's inherited from its parents, ma'am.
Yes, if even you have felt that some of your characteristics, some of your features are similar to one of your parents, right? So how these similarity actually taking place? Because of something which is actually passing from parents to the children, right? So in this chapter, we will try to learn about the inheritance, how the characters are actually inheriting from parents to children, from one generation to next generation. Okay? In your last class, I mean last academic year, you all have studied about the characters, the traits. There are two types of traits, inherited traits and acquired traits. So what is inherited traits? All those characters, all those traits which is actually passed from parents to the offsprings or the children. That is inheritance of characteristics. So those kind of characters are called as inherited characters. For example, your height, your complexion, your hair type, right? Your eye color, the size of the leaf or the leaf color, the fur of this animal, eye color. So these are the inherited character which is transferring from parents to offsprings, from one generation to another. Whereas some of the characters we acquired from the environment, which is not there in the body from the time of the birth. Okay, we are getting these kind of traits or the characters because of the environmental thing, because of the environmental factor, or willingly if we are adapting, willingly if we are using that character, developing inside our body, that kind of traits or the character is called as acquired. Okay, it does not pass from the parents to children. For example, pink color hair. Pink color is not the inherited character. Either it will be black, brown, blonde, right? It depends upon the geographical region. So in Indian geography, the hair color is like black, brown, right? In between the black and brown, but not the pink. But you all have observed in your nearby, in your surroundings, some of the people have a blue color hair, right? Green color, highlighter. So these are what? These are acquired traits, okay? Scars.
Ma'am?
Hello?
Ma'am, this is Anthony, ma'am. I have a doubt, ma'am.
Yes.
Ma'am, what about the children getting white hair, ma'am?
White hair.
In the early stage?
Early stage. So there are two reasons. Either this one is the congenital abnormalities, okay? Or this is the acquired abnormality. There are two things. So this is not a normal trait. This is the abnormality. Whether it is a congenital, okay, or whether it is an acquired one. Are you getting me, Anthony?
Yes, ma'am.
Okay. Scars, again, this is the acquired. Ear piercing, nose piercing, right? So these are what? Acquired traits, which is not coming from one parent to, which is not coming from one generation to next generation. Okay, now look at here. Do you all know who is it? Don't know? Okay, so he is a very famous Bollywood actor, Hrithik Roshan, and he's again a very famous Bollywood actor, but from the time of 80s. Rakesh Roshan. This Rakesh Roshan is actually the father of Hrithik Roshan. If you will see them, they have a very similar features. If you will see the complexion, the eye color, right? So here basically this image is representing that how you are somewhat similar to your parent, but you are not exact Xerox of your parent. Why? Because the reproduction in human is what? Sexual reproduction, means combination of father and mother, and that's why you guys are having some of the features from your father, some of the features from your mother. Though you are getting features, characters, traits from your parents, still you have some uniqueness. You are not exact copy of your one of the parents, right? So let's see. As this is the introductory class, so here we will try to decode some basic terminologies related to the genetics. Okay. So the first term is phenotype. Phenotype means physical appearance. If I will say the physical appearance means what you are able to observe immediately in any of the organism. For example, here the color of the flower, pink color, white color, red color, this is what? Phenotype. So phenotype is what? Physical appearance. Genotype means the trait which is not observable, but those genotypic character or the combination who is responsible for that particular physical appearance. For example, capital B is the genotype. Okay, Nikita, no problem. But please make sure that in the next session, the same problem will not continue, okay? Yes. So phenotype, physical appearance, and those factors, those combination, those traits or the thing which is responsible for any particular phenotype is the genotype. So how you will remember? Phenotype, phi physical, genotype, G gene. Okay, genetic makeup is the genotype, physical appearance is the phenotype. Trait. What is trait? What is trait? You all have studied in your class 10th. What is trait? Yes, and Moldy. What is trait? What is trait, Unmoldeep?
Inherited property.
Very good. Inherited character means the height, means the color. Okay, it's called trait. If we will go specific, means tall or dwarf. This will be the character. Are you getting me? Height. Generally, general term height is the trait. Color is the trait. Okay? And more specific, what type of trait? Tall, dwarf, red color, pink color, okay? Round shape. These are what? Character. So, trait is the inherited character. Inherited character. 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 What is character? Character is a physical property. Okay? Physical property. Sorry, I think I said something wrong. Trait is more specific that if the parent is tall and the genotype, the tallness is inheriting to the children and showing the tallness, right? So this is what trait. The height is the character, physical character. If you will talk about the height, general height is the character. Sorry, I said vice versa. Trait is the specific one, tall or dwarf or any specific color, and character is the general, the physical property. Sorry for that. Okay. So here we will give the example as tall, dwarf. Here it's height. Okay, color, the general one. Now, gene or Mendelian factor. Why here we are saying gene or Mendelian factor? Because when the Mendel was performing their experiment, by that time, he never used the word gene. He always said that something is there, some factor is there who is actually passing from one generation to next. He never used the term gene. Okay? But after the Mendel, Johansen came and he said the factor what the Mendelian was saying is actually gene. And that's why it is written here as a gene or Mendelian factor. Are you getting me? So what is gene? Gene is a specific segment of the DNA which is present inside the chromosome and controls the expression of any character. For example, tallness. Tall. This character, this trait will be expressed by specific gene for the tallness, specific gene for the dwarfness. Okay? So who gave the term gene?
Ma'am, can you repeat again the definition of genes?
Okay. Gene is a unit of inheritance. Gene is a unit of inheritance, which is a segment of DNA, found in chromosome and controls the expression of character. The term gene was given by Johansen in 1909.
Ma'am,
Yes.
ma'am, you told about genes, right, ma'am? Can you
Yes.
repeat again, ma'am?
The whole definition?
No, ma'am, before the segment thing you told, right, ma'am?
Gene
Segment.
is a unit of inheritance. It is a small segment of DNA, which is
Okay.
found in chromosome and controls the expression of character.
Okay, ma'am, got it, ma'am.
The term gene was given by Johansen in 1909. Earlier, it was called as a factor. Now, the next term is alleles. This allele is actually the molecular form, we can say, of any gene. For example, the gene for tallness, which means the gene is made up of allele. Okay, and the term allele was given by Betson in 1905. This allele is also called as an allelomorph. Actually, allele is the abbreviation of allelomorph. So here we said that gene is actually controlling the expression, right? But in gene, alleles are there, and this allele actually showing the or expressing the character. For example, the gene for pink color. Okay, the gene for pink color is BB. This is one gene, and this gene is made up of same kind of allele. Clear? And this allele, the same kind of allele which is present in a single gene are found at the same place or the locus on chromosome or the homologous chromosome. For example, pink color. For the pink color, the gene is capital B, small b. So here there are different alleles. Okay, capital B is one type and small b is the another type. So this gene is made up of different alleles. Clear? Locus. Locus is the fixed place in chromosome. Locus is what? Fixed place, fixed position on a chromosome, which is occupied by gene or allele. Clear? Locus is what? Fixed position which is occupied by gene or allele. Any one of allele. Clear? Prashanti, what is locus? Prashanti, what is locus? Hello, Prashanti. Prashanti, are you there? What are you doing? What is locus, Prashanti? Oh, very good. I thought we are talking about morphology. It is genetics. Is this the definition of locus? Yes, Prashanti. Is this the definition of locus? Jason.
I mean, it is a fixed position of a chromosome where the genes or alleles is present now.
Very good. Yes, Anthony. What is the difference between trait and character?
Ma'am, inherited character is in trait, ma'am, like height, that dark and tall thing, ma'am. But in character, like color and...
The general one.
Yes, ma'am.
Okay. Now, dominant factor or dominant allele. So, any factor or any allele, if they expresses themselves completely, whether in the condition of homozygous or in the condition of heterozygous. So this is wrong. It should be heterozygous. Okay. Heterozygous means the gene is made up of different allele, pair of different allele. Okay. The capital B and small b, both are different. Don't think that these two are not different but the same letter. Though they are same letter B, but still one is capital and one is small, which is representing the same character but different allele. That's why here we used to use same letter to represent single character or the single trait, not character, trait. Clear? So dominant factor or dominant allele means if any factor or allele expresses themselves completely in both the condition homozygous as well as heterozygous, that kind of allele or factor is called as dominant. Recessive, so those factor who get suppressed, whose expression gets suppressed by the dominant factor is the recessive. And recessive factor can express themselves only and only in homozygous condition. So now tell me, which allele is dominant here and which allele is recessive here?
Now, I'm kind of sure about recessive factor again, ma'am.
Recessive factor who gets suppressed or masked by the dominant factor and can express themselves only in homozygous condition. Any allele express themselves when they are homozygous. So here the capital B is representing as a dominant factor. You can see in the homozygous condition, the B color representing the pink color or the red color. If it is in a homozygous, then also they are expressing their color. If it is in a heterozygous condition, then also it is expressing their color. But small b representing here the white color, right? So only in homozygous condition, small b is representing their color white, which means recessive is getting masked by the dominant one. Clear? Is it clear, everyone? What is dominant factor? What is recessive factor? Okay. Now hybrid. Hybrid, I think everybody knows. What is hybrid? Kevin, what is hybrid?
The formation of a new organism or different organism.
Sorry?
The formation of new organism or different organism by crossbreeding someone.
Is it? So are we hybrid?
No. Like another breed.
Moldy, what is hybrid?
In plants, we take two different characters and correlate them. Then this is called hybrid.
This is called hybrid. Okay. N, what is hybrid?
Crossing between two different traits or alleles is known as hybrid.
Sorry?
When there's a cross between two different things, then it is called as hybrid. It can be two different traits.
This is process, hybridization, and the organism produced by this process is called hybrid. Organism is called hybrid and not the whole process is called hybrid. Okay, the end product is hybrid. If you are crossing rose plant and for example jasmine plant, this crossing is called hybridization, and the outcome, the end product, what you are getting after the crossing between these two, that product will be called as hybrid. Okay? Okay, Kevin and Moldeep, you both were saying correct, but the word what you are using for the process was the wrong. Okay? End product is the hybrid and process is the hybridization. Okay? Okay. Next is homozygous. Yes, homozygous means, what is homozygous? What is homozygous? Homo means single. If any gene containing single type of allele, then it will be called as homozygous. And homozygous is most of the time the pure breeding. Sorry, the pure breed, pure line. Okay, hetero means different. If any gene is containing different allele, then it will be called as heterozygous. Clear?
Ma'am, if it's a pure breed, it is called as homozygous, ma'am.
Pure breed are always in homozygous condition. Means the gene will have single type of allele. As you can see here also, for the red color flower, the gene is capital R and capital R. For white color flower, the gene is small r, right? These are the homozygous. Okay?
Yes, ma'am.
Now you can see in the next generation, F2, you will find that The genotype of one of the pink flower is having capital R and small r. Means gene is having different types of allele. And this is called heterozygous. They are pure line or true breed. F1 generation means first filial generation. Once you cross the pure line, once you cross the pure breed, true breed, parent, okay? So after crossing the true breed, first generation will be called as first filial generation or F1 generation. All the organisms, all the offsprings of F1 generation, they have a same type of character. For example, the F1 generation of the pure line of purple, pink or white color, they are showing pink color flower with the genotype capital R, small r. So that means all the offsprings of F1 generation will have the same phenotype, will have the same genotype. Okay. Now, F2 generation. F2 generation is the second filial generation. This second filial generation will be produced by the self-pollination in F1 generation. Offsprings produced by the self-pollination in F1 generation are called as F2 generation. And in F2 generation, offsprings will have different phenotype as well as different genotype. As you can see in the image also, in F2 generation, you will find some of the offsprings are showing the pink coloration and some of the offsprings are showing white coloration. Some of the offsprings having genotype as a capital R, some are having capital R, small r, and some of the offsprings are having genotype small r, small r. This is called second filial generation.
Ma'am?
Yes?
Ma'am, can you repeat F1 generation?
F1 generation is the generation or the offsprings produced by the crossing between pure line, true breed, parent, pure line or true breed parent. It is not mandatory that always the parent will have a pure line. Okay? Are you getting me?
Yes, ma'am.
As you can see, for the F2 generation, the parent is what? Heterozygous condition, right?
Yes, ma'am.
So it is not necessary that always the parent will have a pure line. Clear?
Okay, ma'am.
Genome. What is genome? What is genome? What is genome? What is genome? Yes, Sarjana, what is genome?
Ma'am, it is something related to gene, ma'am. Like,
The name is genome, so something related to gene.
ma'am, like, can you give a hint, ma'am?
No.
Ma'am, Anne answered the chat box, ma'am.
So genome means entire genetic set of any organism, whether the prokaryote, whether the eukaryote, whether the viruses. Okay? Some of the organisms are diploid, so their genome is what? Diploid. Means complete set of gene, genetic makeup. Okay? Some are haploid. Clear what is genome? Very good. Now, Gene pool. What is gene pool? What is gene pool? What is gene pool? Gene pool. Okay, first tell me what is swimming pool? What is swimming pool, Kevin?
A place where water is filled, ma'am.
Collection of water at one place, right?
Yes,
So
ma'am.
gene pool is the collection of gene, aggregation of gene, or the alleles which is present in an interbreeding population. Are you getting me? We all are same breed, human, Homo sapiens, right? So our gene in a collective way is called gene pool. Okay, if any organism in a population doing the interbreeding, so that collection of gene or the allele is called gene pool. Gene pool, sorry. Clear what is gene pool? So write down the definition. The aggregation, or in a better way, it will be like aggregate of all the gene and their alleles. Aggregate of all the genes and their alleles present in an interbreeding population is called gene pool. Bhuvana, can you please read what is the definition of gene pool?
Ma'am, am I audible? Ma'am?
Yes, Bhuvana. Yes, Bhuvana. What is gene pool?
Of all genes and their alleles.
Hello.
Ma'am, can you hear me?
Yes, Bhuvana.
Aggregates of all genes and their alleles.
That's it.
Yes, ma'am.
That means aggregate all genes and alleles within the whole living world.
Ma'am, just now I joined, ma'am. I did not hear you fully.
Okay. Yes, Anmoldi, what is gene pool?
Genes and their alleles present in an interbreeding population is called gene pool.
Very good. Aggregate of all the genes and their alleles present in an interbreeding population. Don't forget to mention the keyword, the interbreeding population. Okay? This gene pool, you will go through this term in while you were studying about the evolution or the ecosystem. Are you getting me? In all these chapters, you will find the term gene pool.
Ma'am, can you explain gene pool again, ma'am?
The gene pool is the collection of gene, okay, whole gene or the entire genome, okay, which is present in the interbreeding population, not between the two different community, between the two different group of population. Are you getting me? We never include as a gene pool the gene present in humans as well as in any prokaryote. No, because they cannot interbreed, right? Are you getting me? Any organism, any group of population who can interbreed, we will include all those genes in a gene pool. Are you getting me, everyone? Okay. Yes, true breeding line. What is true breeding line? Entire set of DNA. What is entire set of DNA? DNA is only one. But in one DNA, there are different types and n numbers of genes are there. Okay, and so we never say that genome is the entire set of DNA. Because throughout the trillions of cells in your body, you are having same type of DNA, right? And in the single type of DNA, there are n numbers of genes. So genome is the entire set of gene and not the DNA. Okay. True breeding line. What is true breeding line? True breeding. What is true breeding or true breed? Please tell me what is true breed.
Self-pollination between two homozygous alleles.
Sorry.
Self-pollination between homozygous alleles.
Self-pollination.
Oh, cross-pollination, sorry.
Yes, homozygous allele is the true breed. Okay. Clear? Who said that? Sarjana. Yes, Sarjana. Very good. So the terms related to genetics are clear to everyone. Throughout the chapter, you will find these terminologies. So I'm not going to explain these terms again and again. So you have to memorize the meaning of each term. Okay. Now, Mendel's contribution. You all have studied about Mendel. You all are aware about Mendel, right? Right? Yes. So who will say? Jason will tell me something about Mendel. Yes.
The homozygous allele is the true breed, and the cross-pollination between two homozygous alleles is true breeding line.
Yes, the process is the breeding. Okay, here the term is true breeding line. That means here the term representing the individual, not the process. Okay, and if any individual is having gene with the similar type of allele or the homozygous allele, there is no mixing. Okay, single individual is not having any mixed, any combination of any trait. Okay, that individual will be called as true breeding line or true breed. Clear? And yes, Jason, tell me something about
Ma'am,
Mendel. Yes.
Mendel breeded three plants, ma'am.
Sorry.
He breeded three plants, ma'am, and he developed three principles of inheritance.
Okay.
Three principles of inheritance describes how transmission of genes in pea plant.
Okay. Anmoldeep.
Mendel is known as the father of genetics. He gives a theory of inheritance.
Okay.
and posthumously he is known as father of genetics because when he researched about inheritance, no will see his research because of evolution theory, and after his scientists were see his research, and he gave his nickname of genetics.
Okay. N. Hello, N. Am I audible?
Yes, ma'am.
Yeah.
Mendel, as she said, is the father of genetics. He conducted many experiments with garden plant. He crossed between different varieties of it to find out how genetics or how the traits are inherited from their parents. He used pea plant because it has many different characters and traits. He made some law. He after seeing all of his research, the results were like he came with 3 laws. These were called as laws of inheritance.
Yes. So the full name of Mendel is Gregor John Mendel. Okay. This Gregor John Mendel is also known as father of genetics. And why he is called as a father of genetics? Because he was the first person who demonstrate the mechanism that how the characters transmit from one generation to another. Okay. Actually, he was not a geneticist. Okay. He did the masters in botany and physics, and then he was the teacher of physics, and then he left the teacher's job and he became a father in one of the monastery or in the church. And while he was a father in one of the church, in his garden he used to grow different plants. So what he did, he was cultivating the garden pea plant. He saw that the pea plant produces uh, 2 types of seeds. Then he was shocked, as he was a botanist. So he got interest and he was trying to find what are the reasons behind this, how the single type of plant can produce 2 different types of seeds. And then he started to collect variety of garden pea plants. So first of all, he collected somewhere around 34 variety. Out of that, he selected the 22 breed, the variety of garden pea plants. And then finally, he came with the 14 variety, which is called as 7 pair of contrasting character or 7 pair of contrasting variety. Clear? So he selected 7 pair of contrasting character of garden pea plant and he started his experiment. Okay. So this Gregor John Mendel, how he started, how he demonstrate that this is the mechanism of inheritance of any character by the process of hybridization. So what he did, he hybridized the garden pea plant. And how he can do this process? Because at that time, the technology was not that much advanced, right? So in your second chapter, sexual reproduction in flowering plant, you all have studied about the artificial hybridization, right? Natural hybridization is cross-pollination. Artificial hybridization, anyone? Remember? No? Ragul will explain. Yes, Ragul, what is artificial hybridization? Artificial hybridization, Ragul. Yes, Ragul. Yes, Ragul. Ragul, I'm waiting. Say yes or no. If you don't know, then say please. I don't know. And then I will give you slap from here only. This time though, I will not accept your excuse. I don't know. Prashanti, artificial hybridization. What are the different steps? Ragul, don't think, oh, bach gaye. No. You are not safe. After Prashanti, you have to give answer.
Hello, ma'am. Can you hear me?
Yes, Prashanti.
Ma'am, the steps are selection of parents,
No. Selection of parent is the first step. After that, the second step is artificial hybridization. And in the second step, artificial hybridization, there is again some more steps.
emasculation, and bagging.
Okay. Now, Ragul, explain me what is emasculation.
Ma'am, removal of anther from a flower.
Okay. What is bagging?
Covering the anther with
Okay.
ethylene bag. Ethylene bag.
Sure.
Yes, ma'am.
Okay. What is dusting?
Dusting. It is the step where the pollen grains are dusted at the anther.
Okay. What is tagging? Yes, Ragul. What is tagging? Arey bhai, aapne you remove the male part from the bisexual flower, right? And then cover the female part so that the female will not be able to get fertilized with unwanted pollen, right? Then you have to select the desired pollen grain, right? And then after the selection, you will dust the desired pollen grain, that is dusting. Once you dust the desired pollen grain, again you will wrap the female reproductive organ of the flower, right? And you will tag that, okay, we are trying to hybridize this and this variety. That is tagging. Okay. And you used to give some time for fertilization, and that is cross. Okay, so this is all about artificial hybridization. Clear? So he conducted the experiment by the process of hybridization. So once he hybridized the variety of garden pea plant, and then he get to know that, oh, there is something who is actually passing from one generation to next generation and passing the information, and that is factor, and that factor is called as Mendelian factor. So Mendel selected 7 pair of contrasting character, and what are they? The height of the plant or the length of the stem, flower position, the inflorescence, whether it is a terminal or at the axis, that is axial, pod color, pod shape, seed color, seed shape. So how you will memorize the 7 contrasting character? By Hi-Fi Shilpa Pandey. Okay, suppose Shilpa Pandey is your friend and you are giving Hi-Fi. Okay, so here height, the length of the stem. Phi means flower color, flower position. There is only 2 things: flower color, flower position. No flower shape. Okay, flower color, flower position. Shilpa means seed color, seed shape. Pandey is pod color, pod shape. Okay, so Hi-Fi, everybody, Hi-Fi. Yes. So please don't forget these 7 contrasting characters. This is very, very, very important for your board exam as well as NEET. Okay. Sometimes in question, the options will be 7 pairs, okay, or 14 characters. So don't get confused with the terms, okay. So he selected the contrasting character, and one by one he started crossing them. So first he selected the height, length of the stem. Okay. If you are selecting single character, okay, if you are selecting single character, that is height or the color, and crossing, that type of crossing, that type of hybridization is called monohybridization, and the output, the product will be monohybrid. Mono means single. Okay, crossing between single contrasting character, whether it is a height, whether it is a shape, whether it is a color. Clear? So what he did, he performed monohybrid cross. Okay, and what is monohybrid cross? Crossing between single pair of contrasting character. So for example, here the character is length of the stem. So one of the trait is tall and another one is dwarf. He selected true breed. Okay, true breed for height, tall and dwarf. Capital T represents tall and small t is representing here as a dwarf. Okay, and then he crossed them. So this is the F1 generation, first Field generation. So in the first Field generation, all the offsprings will have same type of genotype. It is a mixture of both the parent, both the genotype, capital T and small t. Then he did the, he allowed the self-pollination in F1 generation. So the self-pollination in F1 generation, after that what will happen? They will produce F2 generation, second Field generation. Here the offspring, some of the offsprings will have capital T, some will have capital T, small t and small t combination. So this is the true breed and this is F1 generation. All the plants were tall. The phenotype will be tall and the genotype of all the plant, all the offsprings of F1 generation will have capital T, small t. So in a self-pollination means this, what will happen? Capital T, the combination will be like this, will be like this, will be like this, and will be like this. So here the genotype, same type of genotype is, the two offsprings will show similar kind of genotype. Only one offsprings will have capital T combination and one will have small t combination. Now what is the genotypic ratio? It will be 1:2:1. Okay. Now what will be the phenotypic ratio? So this capital T, the pure line itself showing tall. So this offsprings will be tall. These two offsprings, the heterozygous condition will also show tall. Only the small t will be the dwarf. So phenotypic ratio will be 3:1. The ratio between tall and dwarf organism. Till now it is clear? Okay. So the crossing, the monohybrid crossing he did, and what he observed that something is passing from parent to generation. And that's why the first generation is having tall offsprings and not the intermediate. Or for example, if it is a green color or this is a purple color. So offsprings, all the offsprings are green color only. That's how he, by observation, he start formulating the law of inheritance. So what was the observation? Number one, something is passing the information from parent to offspring. That was the first observation. Please write down. Okay, something is passing the information from parent to offsprings. Then second observation, one trait of the character is not appear in F1 generation, but it get expressed in second generation. One of the trait is not getting expressed in F1 generation, but they are showing their expression in second generation. So on the basis of his observation, he said only one parental trait expresses in F1 generation and both the traits expressed in F2 generation. And then he conclude the result that the something which is passing the information is actually the factor. Okay, and some of the factor is dominant And that's why that factor is only getting expressed and other one is not able to get expressed. So all those factors who is not able to express themselves is the recessive one. And that's how he formulated the laws of inheritance. Okay. So the significance of his experiment is what? Laws of inheritance. So by the monohybrid crossing, he gave two laws of inheritance, law of segregation and law of dominance. Okay, so out of all these three laws, law of dominance, law of segregation, and law of independent assortment, which one is the first law? No, Sarjana. The first law is law of segregation is the first one. Because dominance means after the crossing, in the end product or the offspring, some characters are visible, getting expressed, and those are dominance. But before the crossing, there should be the segregation of gamete, segregation of allele in the gamete. The gamete formation will take place, and after that there will be the fertilization of gametes, right? And that's why the first law is law of segregation. Segregation of allele during the formation of gamete. Don't worry, we will discuss about each and every law. Okay? Till now it is clear to you, everyone? Okay. So while he was doing the crossing, that is monohybrid crossing, he saw that one of the character is getting expressed, and he said the factor who is controlling the expression is some factor, and later the Johansson said that Mendelian factor is the gene. So basically one of the gene controls the character. Okay, clear? So after the experimentation, Mendel said that he did the experiment. These are the laws, but people said no, I am not agree with your experiment. Why? Because at that time the technology was not that much advanced that after researching something, after developing something or formulating something, you will go and publish in your own name or you can patent something. Okay, and he was not able to convince the people, was not able to give logic, mathematical logic. And at that time the scientists used to think that research should be convinced by mathematical logic. But as he was botanist, he was not able to convince their colleagues, okay, with the help of mathematical logic. So people denied their experiment. But after some time, as the Anmol Deep said, three scientists came, and who were they? Hugo de Veris, Carl Corren, and Eric von Schemark. So these three scientists came and rediscovered the Mendel's experiment, and he said whatever the Mendel said was absolutely correct. And they said that as per his experiment, as he was the first person who conducted the experiment and said that something is passing, which is absolutely correct, and that's why we can call him as a father of genetics. So now it is clear. Now the question is, why he selected garden pea plant only? Yes. Is there any specific reason
They
for
are
the
easily
selection?
cultivatable, ma'am.
Sorry?
They are easily cultivatable.
They are easily cultivated. Okay. Any other reason?
Ma'am, they showed different characters, ma'am, like height, flower color, flower position. So it will be easy to identify and like differentiate.
Okay.
The
So
pea
number
plant was an
one
annual plant.
sorry,
The
yeah,
pea plant was an annual plant, so it
yes,
can be studied the whole year.
yes. So the reason number one is pure variety of pea plant were available at that time, and single plant have the ability to show variety of character, contrasting character, n numbers of contrasting character you can easily observe in the pea plant. Number three, it is the annual plant with the short lifespan. So you, within a very short duration, you can observe the whole life cycle of pea plant. Plus, you can study throughout the year without getting any disturbance from the nature. Then, pea plant have the ability to produce large number of seeds per plant. And very most important point is pea flower is normally remain closed so that it can undergo self-pollination. But if you want to go for the cross, definitely you can do the cross-pollination. But naturally, they can do self-pollination. So these are the reason for the selection of garden pea plant. So while we are crossing any plant, okay, why we are crossing? Just to get the desired character. And how we will get to know? We need by doing the calculation, right? So here the Punnett, who explained that let's make the whole calculation more easy by the Punnett square. So what is Punnett square? The diagrammatic representation of crossing. Okay. So this is the square. Okay. So this is parent 1, parent 2. And after the segregation, the gamete will have either one of the allele. So let's arrange the male and female allele accordingly. And then now it is quite easy to cross or to fertilize in the form of diagrammatic representation, that capital A, small a, the combination will be like this. And this method, this representation is called Punnett square. Clear? So whenever we used to cross, okay, if you want to know the ratio or the phenotype, or if you want to explain any example, we always use Punnett square to represent the crossing. As this square was given by Reginald C. Punnett, so it is called as Punnett square. So how Mendel came to the inference, the conclusion that this one is passing and that's why this character is getting expressed. This is the dominant one, this is the recessive one, by different crossing method. One of the method to check Okay, whether the crossing or expression what I am saying is correct or wrong, that is test cross. Okay, so what is test cross? Test cross we used to check, or we used to get to know the genotype of the parent. I am not talking about the pure line, any parent, either the homozygous or the heterozygous. So how we will get to know? Suppose in the question, it is given the genotype or the phenotype of offspring, and you get to know, you have to identify the genotype of parent. So how you will get to know the genotype of parent from the genotype of offspring? We will use test cross. So test cross performed between the dominant phenotype and recessive parent. Okay, you already know the phenotype of offspring. Okay, you already know the phenotype of offspring. Suppose two contrasting characters are there, purple flower and white flower, right? We know that purple one is the dominant, okay, and white is the recessive. So what we will do, we need to know the actual genotype of parent, okay, because parent can be the homozygous or heterozygous. So let's see the offspring, which is the dominant, that is purple. It can be homozygous or it can be heterozygous, right? So how we will get to know that the genotype of parent? By crossing these two type of genotype with the recessive one. For example, heterozygous condition of the dominant offspring with the recessive parent. So recessive will always be in a small letter. So small b, small b. This is the first cross. So from this cross, we get to know that whether the parent will be heterozygous, or by crossing the homozygous offspring with the recessive parent. So from these two cross, now it is confirmed that the dominant parent is heterozygous. Is it clear? Shall I give you one question? Yes? Okay.
Ma'am?
So,
Ma'am,
Ma'am?
in the given slide, I think the Punnett square is exchanged for the thing.
are you able to see the slide?
Yes, ma'am. For homozygous, for capital B, the given Punnett square is like capital B, small b.
Okay. Yeah.
The squares are exchanged.
Yes. So let's make it heterozygous only. Okay. And this one is homozygous.
And I think the squares are the only thing exchanged. The below statement is like for the given above one.
Yeah. Okay. Shall I give you a question on this? So the offspring, we know that the phenotype and the crossing between the phenotype, dominant phenotype and recessive parent is test cross. And the phenotype of the offspring is suppose tall. So how we will represent the tall? Whether that capital T or capital T small t combination, right? Now the question is, what is the genotype of parent? Let's see who will give me the answer. Genotype, offspring is tall, okay? So you can imagine whether the genotype is capital T combination or the capital T small t combination. Now you have to identify the genotype of parent. What will be the genotype of parent if the offspring is...
Ma'am, can you repeat the question again, ma'am?
If the offspring is tall, what will be the genotype of parent? Done? Is it done, N? Do the crossing in your notebook. Because whole chapter is about crossing. Okay, and Sarjana, Bhuvana, Kevin, everybody is saying the genotype is capital T. Okay, now let's see. The phenotype, dominant phenotype is tall. So here, the genotype may be the capital T, small t. Now let's cross this capital T with the recessive parent. So what is the recessive? Let's cross capital T and small t. This is first, okay? Now let's see. As per homozygous condition, parent is heterozygous. Now let's see another condition, heterozygous. So in heterozygous condition, capital T, small t, let's cross it with the recessive one. Now, what will be the answer? Capital T, small t, capital T, small t, small t, small t, small t, small t. So what is the genotype of dominant parent? Yes. And Sarjana, Bhuvana, Kevin. What is the genotype? What will be the answer? Why you people said capital T? Please tell me the logic.
Ma'am,
Yes,
you
Sarjana.
said that all the offsprings are tall. If the parent was like capital T, small t, you also get a 50% chance that it can be recessive because you're doing a test cross and it is with the recessive gene. So the offsprings can only be tall if the unknown parent gene is capital T.
Why? Capital T, small t combination cannot express.
It can express, ma'am, but there is only 50% for the possibility. But in capital T, there is a 100% possibility.
Yes, that is. But as per crossing, the answer will be heterozygous because you cannot go with the possibility. Okay, by crossing you have to give the reason. By crossing you have to give the answer. Are you getting me? We know that if the capital T combination is there, which is the 100% chance, 100% surety that the plant will be tall, right? So actually, what is the genotype of parent? So we have to go through both the condition homozygous and heterozygous, because the dominant factor can express themselves in both the condition. And it is clearly said that the phenotype is dominant. And you have selected the genotype, one condition homozygous and another condition heterozygous. And for the test cross, we have to cross the offspring dominant phenotype wala offspring with the recessive parent. While crossing, whatever the genotype you will get, this is the actual answer of unknown genotype of unknown parent. Okay, are you getting me? What you are saying that capital T will be the answer is the theory. Okay, but logically this is not true, because the dominant factor can express themselves completely in heterozygous condition also. Are you getting me, N?
Yes, ma'am, but according to the question, aren't all offsprings tall?
I said offspring is tall, right? And we have to identify the parent with the help of test cross. So how you will do the test crossing? You have to check both the condition now, homozygous as well as heterozygous, because we know that dominant can express themselves in both the conditions. And whatever the genotype will come is actually the answer to this question. Chalo, I am giving you one more example. I mean, one more question. Okay? So
Ma'am, so for this question, we have to like do the cross, and
exactly.
we have to.
Yes.
So we have to write the statement at last, no, ma'am. Like there's possibilities like for both the type, no, ma'am.
Exactly. If you are doing the test cross, you have to show both the condition, heterozygous as well as homozygous.
Okay.
Otherwise, your answer will not be accepted by the examiner.
So
in
we
board
cannot.
exam, okay?
Yes, ma'am.
In NEET exam, it is fixed, because the examiner knows that by crossing only we will get to know the answer. So if it is about the descriptive question or the MCQ, please always go with the crossing. Whatever the result will come from the crossing, that one will be the actual answer. Okay. Chalo, one more question. The shape of seed, okay, of offspring is round. Now you have to identify the genotype of unknown parent. If the seed shape is round, what will be the genotype of unknown parent? Round R. As it is a dominant, so let's make it capital R, capital R. And for recessive, it will be small r, small r. This time, Jason, Madhumita, Prashanti, and Anthony will answer first and then other one. Yes, Akhilesh, you can cross. This time, along with the genotype, you have to tell me the phenotype of unknown parent also. In question, always be there a recessive parent, okay? And recessive parent cannot be heterozygous. They always be in homozygous condition only. So one is fixed, the homozygous. Yes, Jason, what is the answer? First tell me phenotype.
In phenotype,
Of parent, unknown parent.
there's a 100% chance of oval,
100% chance of
oval seed to produce.
oval, round.
Okay, sorry, round. On the other side of the
In
crossing,
in what condition there is 100% chance that parent will have a round shape?
in crossing homozygous dominant phenotype with recessive parent.
If we cross homozygous offspring
With recessive parent.
with recessive parent, okay. So homozygous offspring means let's say capital R
Yes.
with recessive means small r. The
Yes,
answer
ma'am.
will be capital R and small r. The phenotype will be round. Very good. Okay. Another condition?
Now the next condition of crossing heterozygous parent heterozygous offspring with recessive parents.
Okay. So what will be the phenotype?
There's a 50% chance of round and a 50% chance of not round.
Not round, then what? Wrinkled.
Wrinkled.
Okay. There
Smaller,
are two.
smaller.
Yes. The contrasting character for seed shape is round and wrinkled. So everybody got the same answer? Okay, very good. Another kind of cross is back cross. So what is back cross? What is back cross? Test cross, though we know what is test cross. What is back cross? Yes, anyone? I think test cross and back cross is there in class 10th also. Yes, what is back cross? Selfing of F1 generation. Oh, what is back here then? Yes, Akhilesh, you only explain me what is back cross. Yes, Akhilesh. Selfing of F1 generation, Sarjana is saying with the dominant parent. Okay, that means in a test cross, we are crossing the dominant offspring with the recessive parent, and in back cross, we will cross the dominant offspring with the dominant parent only. Is it? Yes, Sarjana.
Ma'am, it's not the dominant phenotype with the dominant parent itself. Doing, I don't know the first one, but it's something related to the dominant parent. There we took recessive, we are replacing here with the dominant parent.
Okay, so basically back cross means we are crossing with the back generation, previous generation. Okay, that means the cross of F1 generation with any one of the parent. If we cross F1 generation with any one of the parent, whether the dominant or the recessive, then such cross is called as back cross, because the current generation, we are crossing the current generation with the previous one. Okay, this is called back cross. So again, there are two possibility, because there are two kind of expression, whether it is a dominant one or the recessive one. So if we cross the F1 generation with the dominant parent, or if we cross the F1 generation with the recessive parent. So there is nothing like a test cross that it is fixed that one of the dominant phenotype wala offspring we will cross with the recessive. It is not fixed here. Okay, here the thing is we have to cross the F1 generation with any of the parent, whether the dominant or the recessive. So phenotype will be accordingly if we cross the F1 generation, okay, with the dominant one. For example, here. So if the dominant is homozygous, then what will be the phenotype? All the offsprings will be dominant. Okay, they all show the dominant character, but the ratio of genotype will be 1:1, or the 50% genotype will be homozygous and 50% genotype will be heterozygous. Now let's see the another condition. F1 generation. F1 generation will always be the hybrid, means always in a heterozygous. So let's see the recessive parent. So what will be the answer? Means half of the offspring, 50% offspring will show the dominant character. The phenotype will be dominant and 50% will be recessive, right? If we cross with recessive parent. Okay, now let's see the dominant one. Again, there are two conditions. Whether we cross the F1 generation with the pure parent, pure dominant, or with the heterozygous parent. Clear? Is it clear to you what is back cross? The thing is crossing between the F1 generation with the previous generation, the parental generation. Okay? It is nothing like it. The parent should be only the dominant one or only the recessive one. No. Is it clear?
Ma'am,
So
I
why?
have a doubt, ma'am.
Yes.
Ma'am, even in the test cross we saw, we are taking the dominant phenotype and we are taking, like, it can be capital T or small t, and we are crossing it with recessive parent. So capital T small t crossed with small t, and even in back cross it is coming like same, no ma'am, like capital T into small t, like both are.
In test cross,
Yes, ma'am.
we are determining the genotype or the phenotype of unknown parent. We only knows the phenotype of offspring. And from the phenotype, dominant phenotype of offspring, we have to determine the phenotype of parent, unknown parent. And for that, we use test cross. Clear?
Yes, ma'am.
Back cross, it is not fixed, it is not mandatory that you have to cross the dominant phenotype wala offering with the recessive one only. No. The thing is F1 generation. Okay. Crossing between the F1 generation offering with the previous one. We are moving backward. Okay. So we can cross either with the dominant parent or the recessive parent. Here again, the genotype is not fixed that you have to cross with only with the homozygous genotype or heterozygous genotype. You can cross with anyone. Why? Because by the help of this cross, we are going to detect the congenital disease. We are going to detect the inheritance of disease. Whether the genes are having ability to resistance the expression of disease or not in the combination, in the homozygous condition or in heterozygous condition. Just to check, okay, whether the offspring is suffering from any disease, it is related to gene only, it is inheritable or not. How we will get to know? By the help of back cross. Okay, from the offspring we will move towards the parent. Okay, just to check whether the related disease is getting inherited from the previous generation only. Clear? Whereas in test cross we are going to identify the phenotype of unknown parent. You can think that we know this is the kid and we want to know who is the father, right? So this is the one. But the process is different for checking the paternity, which is completely different. But I am just saying the logic. Okay. Another cross is reciprocal cross. What is the meaning of reciprocal? Yes, Kevin, what is the meaning of reciprocal?
Like inversion, ma'am.
Sorry?
Like inversion related to that.
Can you explain me what is reciprocal with the example?
I don't know exactly, ma'am.
Okay. Anthony, what is the meaning of reciprocal?
Ma'am, reciprocal. I don't know, ma'am. Exact meaning. I don't know. Left behind something. Ma'am.
Our parents used to say, she always reciprocates me. Yeah?
Yes, ma'am.
What is that?
Doesn't obey, ma'am.
Sorry?
I don't know, ma'am. Exact meaning. I don't know. Ma'am.
Okay. And what is reciprocal?
And the word meaning we use, like when parents say, means it's like agreeing to someone, but in math it's like if 1 by 2 is there, then the reciprocal is like 2 by 1. I'm not sure which it refers to here in genetics.
The maths one. Okay. So suppose male is having red color, sorry, not red, male is having black color hair. Okay. And the female is having brown color hair in one generation. In next generation, male is having brown color hair and female is having black color hair. This is what? Reciprocal. Are you getting me? एक जनरेशन में ऊपर है तो एक जनरेशन में उसका opposite. Are you getting me? Just like N said in maths, 1:1 in one condition, in second there is a 2:1, right? Yes. So this is reciprocal and this is the reciprocal cross. So look at here. This cross is, we usually do this cross just to know whether the character is sex linked or not. Okay. And the sex chromosome is called as autosome. So sex linked trait is called autosomal trait or autosomal character. So look at here in the image. In one condition, mother is having red eye. Okay. So here we are representing the genotype of mother is W plus W plus. Okay. And the father, the genotype of father is W and Y. Okay. One more thing. In the, as you can see, the female is having homozygous sex chromosome, right? Autosome is homozygous and male is having heterozygous, right? So here the male is called heterogamity condition and female is homogamity condition. So while we were discussing about the sex determination, all these terms will come. Okay, heterogamity, homogamity. Okay, so when we cross the mother with the father, what will be the outcome? In the first generation, we will see W plus with the normal W of the male, right? And the expression will be like red color. As the combination is WW, which means it will be the female. Just like in a human XX and XY, the same. Okay. Now let's see male, W and Y. But the color, eye color of the father is, the son is what? Red. Why? Because of plus. which is linked here. The W plus is representing here the color of I, which is a sex linked. Okay. Now reciprocate. Here the mother is normal W. Earlier the mother was linked with plus, but in another condition, in the second half cross, the mother is having normal W and father is having W with plus. So when we cross, what will happen? The outcome, normal W from mother and W plus from father, they make a combination female with the red eye color. Now the male is having W plus Y. Okay. The W, which is the normal one, but still the son is showing red color eye. So this cross says that the color of the eye is sex linked. Okay, is sex linked because why? It doesn't have any gene or the gene linked with the Y chromosome. Are you getting me? But still son is showing the red coloration, the red color eye. Son is having red color eye. Why? Because this character, this trait is sex linked. Are you getting me?
Ma'am, even the
Yes,
female has red color eye here, no ma'am.
Sorry?
Anyway, it is in the given cross, anyway the progeny is having red color eye. Even the female, it is, they are saying like it is red colored.
Because of the linkage of gene with the sex chromosome, and that's why you can see here the father is having a gene linked with the W chromosome, but Y it doesn't have, and they are showing the white color eye, but the progeny is having red color eye. Why? Because the W, one of the W is coming from the mother, which is having the gene for red color. See, we have reciprocate. In the first condition, here the W is having plus, but in second condition, the father whose genotype W is having plus, right? Though we reciprocate, still wherever the W, the genotype, is coming from the mother, whether in the female or the male, they are showing the red color eye. And this represents that this particular character is sex linked. Are you getting me or still confused? Everyone, please let me know. Because this is completely new term for you guys, right? Reciprocal cross. One character or one allele, okay, allele X, in one condition is with the female genotype. Okay, let's reciprocate. In another condition, this particular X is linked with the male genotype. Okay, when this X was linked with the female genotype, in that condition, wherever this X linked is getting combined or it is present in any of the genotype, whether it is a male or female, they are giving their expression. Once we reciprocate, once we opposite, that is in one condition it was a female, so let's reciprocate. In another condition, this is linked with the male, okay, male genotype. In that condition also, once we reciprocate, that too, this gene is showing their red coloration in both the condition male and female. Okay, and this represents that whether it is present in male or female, this particular gene will express them completely because it is a sex-linked character. It is a sex-linked trait. Now it is clear? Say yes or no. Is it clear or no? No. Okay, chalo. Let's take another example. XX for female, X and Y for male, right? So here the XX is like this. Okay, in one condition, if we will cross, it will be like this. Okay, and in all the offsprings, the gene is expressing themselves completely. Okay, now let's reciprocate. The X is normal, okay, and the male is having linked character. So let's see. Okay, so here the female is showing the linkage, right? That character is sex-linked. Though the male chromosome, the male genotype is showing that it is a the character is sex-linked, right? So what will happen whenever we crossed and we got the offspring, the progeny? This particular gene will express themselves, okay? Though we are not representing by P here, but still they will have the expression. But in the next generation, this is about the F1 generation, okay? But in the F2 generation, it may be differ. And that's why this reciprocal cross is not globally accepted for each and every kind of examination for whether the character is sex-linked or not, for all the type of character. Okay, and that's why it is not a very common, and that's why you guys are knowing this term first time, right? Because this is not a common cross. Very rarely this cross is actually used by the researcher. So the test cross, back cross, reciprocal cross is clear to you? Say yes or no. Now let's a tall true breeding garden pea plant is crossed with dwarf true breeding garden pea plant. When F1 plant were selfed and resulting genotype were in the ratio of what? So first you have to see tall true breeding plant. True breeding means homozygous. Is crossed with dwarf true breeding plant, which is again this. Now, in the first generation, all progeny will be capital T, small t. This is the F1 generation. Once we do the once they solve self-pollinate or selfing, which means capital T, small t. Now, what will be the genotype ratio in F2 generation? So, do the selfing of F1 generation and tell me. Resulting genotype. Yes, correct answer will be capital T, then capital T, small t, and then small t, small t. So 1 is to 2 is to 1. Which one? Yes, correct answer is option A. Yes, this one. Now, how many pair of contrasting character in pea plant were studied by Mendel in his experiment? Very good. 7 pair of contrasting character. Test cross is carried out to determine the genotype of plant at F2 generation, predict whether the two traits are linked, assess the number of allele of a gene, determine whether two species or variety will breed successfully or not. Option A, determine the genotype of plant at F2 generation. What is test cross? What is test cross? We know the phenotype of offspring, right? And then we have to know the phenotype of parent, unknown parent. So once we know the phenotype of the parent, definitely we will get to know, first we will get to know the genotype of parent and then phenotype. So from the genotype of parent, definitely we can predict the genotype of generation F2. Very good. Correct answer will be option A. Alleles are what? Different phenotype, true breeding homozygote, different molecular form of a gene, heterozygotes. What will be the answer? Different molecular form of gene. Very good. So in today's class, we had learned about the Mendel's contribution in the field of genetics, monohybrid cross. In monohybrid cross, there are different types of cross by which we can determine the genotype, phenotype, and even the sex-linked trait, the disease, right? With the help of different cross. Then we had learned about the 7 contrasting character which is selected by the Mendel. So always remember, high five Shilpa Pandey. Now, these cross like test cross, back cross, and reciprocal cross. Please go through the statement given here and let me know if you are getting the statements or not. Done? Yes. Prashanti, Kevin, Anthony, N.